Hi, everybody. Thanks for joining me today. And also a big thanks to Labroots for organizing this virtual conference. My name is Ben Borgo, and I'm the portfolio manager for the Genome Engineering and Modulation Group at MilliporeSigma, which is the life science business of Merck KGaA, Darmstadt, Germany. Today, I'm going to tell you about some of the work that we've done in developing immuno-oncology cell line standards and how these standards can be used to improve potency testing in various immunotherapies, for various immunotherapies. So within MilliporeSigma, we have a number of groups which are dedicated to the development of genome engineering tools. And we also have several groups that focus on using those genome engineering tools to create novel cell lines, cell models, and other engineered cells for various purposes. One of these groups, Cell Design Studio specifically, focuses on the types of cell models that are relevant for immuno-oncology research. And most of the work that I'll talk about today was done either with materials produced by Cell Design Studio or actually by our scientists within Cell Design Studio. So as I mentioned, we develop and use a wide variety of gene editing tools. These tools include, but are not limited to, CRISPR-based tools, things like nucleases, as well as CRISPR activation and interference, and CRISPR integration using homologous recombination or HDR-focused tools. It also includes a proprietary nuclease-based editing tool that we use called zinc finger nucleases, which are a little bit older than CRISPR technologies. But we use that primarily to generate models and cell lines that have fewer IP restrictions than something like CRISPR. Works essentially the same way. The same sorts of applications are accessible to zinc finger technologies, but in many cases it's much harder to design. And so the expertise that we've built up with that allows us to do that effectively. So in general, we introduce these tools into cells in a variety of formats, including things like lentivirus, direct introduction of RNPs into cells, using things like nucleofection or electroporation, as well as chemical delivery methods. And we do both internal and customer research projects in immuno-oncology, cell therapy, iPS cells, and gene therapies. As a service, we offer a wide range of fully customized project options depending on our clients' needs. Our focus largely being on being a collaborative research contractor. And so we pride ourselves on being able to take on the most difficult or complex projects and work directly with our clients in order to solve those problems when many other CROs might not be able to do such a thing. So many of the projects that we take on have common themes. We often focus on the earlier stages of research, but we can provide GMP-like process control all the way out in the early clinical development. Our projects range from gene modulation and knockouts to difficult integration and generation of reporter constructs. Really, there's no project too difficult. There's no project too large. We like to, again, take on the things that we find most challenging, and some of the projects that I'll tell you about today fall into those categories. So we take on these projects and try to deliver exceptional results using a number of key contributions. First, we develop and utilize our own unique and proprietary gene editing reagents. So this can be things like CRISPR, where we have an extensive IP portfolio, or they can be things like I mentioned before with the zinc fingers. Many of these become available through our Cell Design Studio well before they can be purchased by external researchers, meaning we provide immediate access to the most state-of-the-art tools via our service offering. Our cell line engineering services make these tools accessible by helping you design your research projects and choose the right tools for the right job. And as I mentioned, we provide full service cell line development across the entire development pipeline. The next thing I want to talk about is a couple of cell lines that we have developed, how those are being used to make immunotherapy development and assessment more standardized and more consistent. The first project I want to discuss is the development of what we call our landing pad cell lines. And so as most of you probably know, selection of model systems for studying, for example, TCRs is often a trade-off between the robustness of the system and the relevance of the biology. So, for example, primary T cells can be very difficult to culture, but are the actual cells that are representative of the biology when you're looking at immunotherapies or how a patient's immune system reacts to something like a cancer treatment. And Jurkat cells, on the other hand, can be grown pretty easily, but they're not the perfect proxy for primary T cells. Being an immortalized version, their underlying biology has been altered, and often the TCRs that they present are not fully representative of what you would see in a normal biological specimen. So what if you could then engineer those Jurkats to express the specific TCR of interest rather than the melange of TCRs that you might get in a standard Jurkat line? This is the question that we asked ourselves as we decided to engineer some of these landing pad lines. And so the basic concept with landing pad cell lines is that we provide a readily interchangeable set of cassettes that are integrated into a variety of biologically relevant cell lines. And so one of those is Jurkats, like I mentioned, and then the others, A549s, A375s, HCT116s, and as on a custom basis, THP1 cells are also biologically relevant cell lines for different cancer types and are good cell models for a number of oncology studies. And so the landing pad insert I'm showing here at the bottom of the slide, where there are several cassettes that are interchangeable using Cre-mediated recombination, which is much more straightforward to use than some of the existing technologies for doing homologous recombination at various genome sites. The cassette is integrated into the AAVS1 safe harbor locus, which allows the integration of individual gene constructs with limited disruption of other target genes in the cell. This simplifies the process, especially for situations where you want to express a single gene construct, like TCRs in this case, in one of these listed cell lines. And it makes it so that you don't have to worry about off-target effects or integration of your construct into sites other than this specific one. And a good example of how we use these is the application I mentioned, which is the introduction or overexpression of a single uniform TCR of interest. We've used these landing pad cell lines to generate TCR expressing Jurkats, where we're able to quickly substitute in any TCR of interest using Cre-mediated exchange. And then we can screen these cells that are expressing the TCRs for T cell activation against HLA presented antigens. So this is just one example, one simple example of where we can, in a uniform and controlled way, introduce specific TCRs into a known population of cells and then either screen those cells or assay those cells, knowing what's on the surface, rather than kind of working the other way around and doing the assay and trying to figure out what was on the surface of the things that we were looking at. And so in that vein, but shifting gears slightly, antigen-presenting cells also provide the same sort of a challenge for consistency of expression of their presented antigens. And so we've similarly engineered several panels of antigen-presenting cells, which provide for controlled levels of antigen expression. So these cells are engineered, again, within biologically relevant cell lines. You can see them listed here on the right, Raji cells, A549s and A375s. This is a distinct advantage over any sort of bead-based or artificial support of antigens that are used to activate T cells, but they do offer a similar advantage of having controlled levels of antigen expression that make them much more versatile for determining things like potency curves or other metrics that require a precise enumeration of the antigen, things like T cell activation or cytokine release thresholds. And so these sort of controlled systems that, again, are still representative of the biology that we're trying to study provide a much better system to understand what's going on in these cell lines. So I mentioned HLA-presented antigens when I was discussing our landing pad lines. We also have developed a full panel of monoallelic HLA cell lines. each of which has had all of its endogenous B2Ms knocked out and the expression of a single HLA subtype introduced via lentiviral expression. So these lines that we have listed here represent over 60% of the HLA types in the general population. And they do, again, provide a controlled system for testing antigen presentation and the associated T cell activation. So now that I've showed you some of these tools that we want to utilize and that we have created so that we can improve testing, I want to go through one application that we worked on with a collaborator as part of an early collaboration using specifically these HLA cell lines. And so the basic idea here is that our client had a suite of antigenic peptides that are known to be presented in the HLA complex. They also knew that patients with a specific HLA subtype generate T cells which actively attack cells presenting those antigenic peptides. And they would like to understand if the T cells generated in these patients, with a specific HLA subtype, also attack the antigen when it's presented in other HLA subtypes. So they sought to understand if T cells activated by that peptide library would be subtype specific. So to answer these questions, they required two things. The first is HLA type T cells or PBMCs from donors that were stimulated with the peptide libraries. That's something that our client generated. And the second thing that they required was those monoallelic HLA cells that would allow them to test the efficiency in a controlled system with which T cells can kill HLA subtype specific tumor cells. And so this is the primary component that we provided within this collaboration. So to set up this experiment, we first verified that the controls behave as expected. Specifically, in the first figure, you can see that the knockout HLA is shown in red as the baseline for no HLA expression. This is stained using HLA antibodies and sorted by FACS. In blue, the shift of the peak represents the presence of a fluorescent anti-HLA binding to our engineered HLA subtyped cells. In the second figure, we introduced the expression of the peptide antigens and showed the same shift, demonstrating that the HLA is still present and it's active with the addition of the peptide antigen expression. In the final figure, we have expressed the peptide antigen in the absence of HLA. And so the library is present, but the HLA is not. And as expected, we don't see surface presentation of the HLA when we stain with that anti-HLA antibody. So this basically tells us that those cell lines that we've engineered behave as expected. They're able to present the HLA complexes and the antigen within the HLA. When the HLA complex is knocked out, and that is part of our HLA panels, the knockout cell lines, those antigens cannot be presented on the surface. And so to test the killing efficiency of T cells, first PBMCs are extracted from HLA type donors. That sample is stimulated with the antigenic peptide library that I mentioned before. Activated T cells are generated that should recognize the HLA presented antigens in a cell-based assay. Each set of the donor T cells is then introduced to our cell panel and all of the controls. And the killing of the cells in the panel is measured both by cytotoxicity and interferon gamma production. Both are measured by staining and sorting by FACS, and both are proxies for the killing activity of the cells recognizing the antigen on the surface. If the T cells are HLA subtype specific, we would expect that they would only kill the cell lines in the panel that match the donor of the T cells, but would not interact with the others. They would not be able to recognize the antigen on the surface of the other HLA subtypes. So that's the basic setup. We did this with a number of different HLA subtypes and a number of different HLA typed donors. So the HLA panel that we've generated covers most of the donor subtypes that this particular client was interested in. which isn't surprising given that our panels cover 60+% of the population. It didn't cover all of the target alleles, so they had some donor PBMCs that had HLA subtypes that sat outside of what we had in our panel. This is something that we could have always engineered as an additional cell line. However, this particular client felt that the representation we got across their HLAs of interest was sufficient for this particular study. And so here are the results from the cytotoxicity and interferon gamma assays performed at our collaborator. So the red bars represent significant killing of T cells as measured by cytolysis and interferon gamma production. Only one of the donors, specifically donor 2 in the second column, had significant killing in an off-target subtype, and that is represented there by the red bars. So the bigger the red bar, the more the killing activity of that specific type, and you can see in most cases, the killing activity of a donor's specific PBMCs is isolated to a single HLA subtype. So T cells generated from a donor with a known HLA type seem to be specific for killing cells of the same HLA subtype with this particular set of antigens. This indicates that something like a cell-based therapeutic targeting these peptides as an antigen would only be efficient if the target cells were of the same type. It would have to be specific for the HLA subtypes. There's one unique example, specifically with donor number 2, where there's some cross-reactivity between different subtypes, but we didn't explore this any further at this point. It seems pretty clear that the trend is, in general, to that specificity. So this level of detail wouldn't be obtainable without those monoallelic HLA target cells. And so having those engineered cells really enabled a controlled study of how PBMCs specifically reacted with this antigen peptide library and how specific donor T cells are for these different antigens. So in conclusion, the primary T cells stimulated with an antigenic peptide library were utilized alongside a suite of monoallelic HLA cells to enumerate the specificity of the T cell response. Single change HLA B2M constructs within these cell lines are functional. They can present peptides to the T cells, and the antigen presentation carries over to a wide range of HLA subtypes. The monoallelic lines thus provide a clean, defined background for assessing this type of T cell activity. And again, this extends not just to the A549 cells that we tested here, but to the JERCATs and the A375s, which make up the other HLA panels that we currently have available. And I would also again mention that if the particular cell lines that you are interested in are not a member of our HLA panels, we have a very robust pipeline for building these types of cell panels. And so looking at other cell types is not beyond the realm of what we would normally do as part of a collaboration. So I just want to close by mentioning again that MilliporeSigma and Merck KGaA Darmstadt, Germany provide a broad range of development and manufacturing services for cell and gene therapies. This includes the cell design studio, which is the primary focus of what I talked about today. All of the materials and projects that I mentioned were either hatched within the cell design studio or hatched as part of a collaboration or project with an outside, with a third party, but involving the work cell design studio. We also offer bioassay validation services and full service CDMO manufacturing for things like cell and gene therapies via our BioReliance services. And we do CAR T manufacturing and testing. And so as a company, we offer an extremely close level of collaboration and partnership. This is something that you might find lacking in other CDMOs. The projects that I showed here are really good examples of how closely we work with our partners. So please do reach out to us if you have any projects on which you believe we can help, questions about the work that we've done here or what our capabilities entail. Otherwise, the contact information for any of the materials or materials or services I mentioned today are listed here, and I'm happy to take any additional questions. Please do visit our website. Some of the other projects that are related to this are listed there. We have a site specifically for immuno-oncology. These cell line panels are mentioned there, as well as some of the other work that we've done with genome engineering tools and with cell engineering projects. So with that, I will close. Say thank you for your attention, and I am happy to take any questions that you have.